the thinking we've been doing recently. So I'm really keen to kind of hear feedback about it and get other people's insights.
Great. Well, let's get you some feedback today.
Great. All right.
I love that the discussion is already going before we've even started. Fantastic.
That's great.
So keep it coming. Hello and welcome, everyone. My name is Stephanie Forker, and you managed to find the clinical neuroanatomy seminars today with James McShine talking about the thalamus. So just briefly, the way it works is some of you have joined us over here on YouTube directly. Some of you are watching over on YouTube, and both teams can ask questions throughout the talk. So if you just want to write your comments and questions in the chat box on Zoom or the comments on YouTube, Michelle and I will pick them up and then relay them to our wonderful speaker today. The Q&A after the talk will be led by Michelle Thibault-Deschotten. And today's speaker was actually recommended to us as a rising star in the field.
Wow.
And
no
following the advertisement on Twitter and what people tweeted about you, that seems to be definitely the case, very well received. So I'm very pleased that you have the time and speak to us today, and to see that so many people have joined us today live on Zoom and over on YouTube. Mac is a Robinson Fellow at the University of Sydney at the moment, and he uses multimodal imaging to study cognitive functions in health and disease and maps brain dynamics. And you already heard that he's keen to talk about it, so I'm not going to steal more of your time and give the floor to you, Mac, to talk to us about how the thalamus integrates the macrosystem of the brain to facilitate complex adaptive brain network dynamics. Take it away.
Well, thank you, Stephanie. What a lovely introduction. It's absolutely my pleasure to be here tonight. I don't know what time it is now in Paris or where you're watching from, but it's getting into the evening here in Sydney. But please, I'll echo what Stephanie said. If you have any questions at any point in time, or you'd like me to go back over something or to dive into detail about anything, please feel free to interrupt. I'm more than happy to field any questions as we go. So the starting point for my talk tonight is this just beautiful video of a larval zebrafish. So there's something quite interesting about larval zebrafish, which is that they're translucent for the first few weeks of life. And it turns out that if you inject them with a particular calcium fluorescence molecule, every time the level of calcium in a particular cell increases, you see a flash of fluorescence, like the fluorescent algae you'll see off the coast of some oceans in the right time of year. And so what that means is that we can directly observe while this animal is held stationary in a gel through which it can breathe. It can breathe through the water that passes over its gills. You can observe its entire brain in action. And I think it's important for us as neuroscientists to constantly come back to things like this, where we can see the system working and see the system functioning in real time, to remind ourselves of some of the interesting features that happen. I'm not going to be able to solve all those problems. I can't describe to you why this neuron lights up at this point in time and another one goes, or why you get that huge flash that you see of this sort of systemic calcium transient. But what I am going to try to do tonight is describe what I see as an organizing principle that might start to give us some insights about how different parts of the brain, different macro systems of the brain, are integrated together, how they connect with one another, how they influence one another, and to think about what some of the implications for that of those connections might be for how the system as a whole can function. So as neuroscientists, we all have our own specialty. And there's a great quote, which I'll probably absolutely butcher, which is that by the time you leave your PhD, you're trapped in your own expertise. And I think that's really true. And I think the reason that that's a little bit harrowing for us as neuroscientists is that I don't think any one specialty that currently exists is going to somehow solve the brain, as it were. There's so many different ways to look at the brain. You can look at it as a spatial system. You could look at it as something quite abstract, like a Bayesian model. You can go in and look at individual synapses down at the microscopic level. You can zoom out and do whole brain imaging like Boulder EEG at the whole macro scale level. Now, none of these systems is the correct one to look at it, but rather the answer for how this system works is some combination, some synergy between those different viewpoints. And so I think one of the things that I'll try to bring out with tonight's talk is a sort of ham-fisted attempt to try to see what some of those intersections might look like between some fields that I think have a really important role to play in helping describe the system. So the starting point for the talk and really the kind of main focus for it, I don't know if the little Zoom bar is in the way. I'm just going to try and move it down here. All right. So my starting point is somewhere that I think as a sort of group of neuroscientists in neuroimaging, we often tend to overlook. But it's actually a really crucially important region, and that's the thalamus. So this is a bilateral structure in the diencephalon. It sits atop of the midbrain, right in front of the superior colliculus and the habenula, tucked in behind the caudate nucleus. You can see the hippocampus and the amygdala wrapping around it. This really, really important structures. Number one, they connect to almost every other part of the central nervous system. And number two, they're crucially involved in many different functions, both sensory, but also emotional and higher order, things like memory, attention, and cognition. We do know a lot about the way the thalamus works. One really beautiful study that was done, in fact, I'll have to move my thing again so you can see who I'm quoting. One beautiful study that was done back in the early 2000s by the group at Oxford used diffusion imaging, only in a small sample, but it was really early days. And they showed that you could actually track a really interesting gradient of connectivity around the thalamus. So if you put a seed in the different thalamic voxels and looked at track tracing, you could actually see that there was a particular logic to the organization of the thalamus, where there's connection, say, between the frontal lobe and areas inside of the mediodorsal nucleus. And as you track backwards, you can see these really punctate areas of higher connectivity between the thalamus and the cortex, which wraps around into the temporal lobe. But the thalamus itself is not just connected to the cortex. It actually has really important connections to many other regions. And another important point is that the thalamus itself doesn't actually have a single mode of connectivity. So if you go back to at least the way I was taught about it in medical school and in a lot of the traditional textbooks, many people focus on what are called the sort of thalamic relay nuclei. So I'm showing you those here in blue. And the idea of this circuit is that there's a little nucleus, let's say a little cell in the thalamus here, that maybe receives a retinal input, and then it projects straight onto the cortex and it hits, let's say, back in V1, and then V1 goes off and does its connection with the rest of the brain. There's also a projection back down to the thalamic nucleus that project to that same zone. And along with that, you see these really important inhibitory neurons. This is the reticular nucleus, which is like a shell surrounding the thalamus, and these interneurons within the cortex that are also very important. It turns out that is just but one of many different ways that thalamic regions project to the cortex. Another very important and probably the other major class of these neurons is actually denoted here in red. And instead of projecting to a very precise region of cortex, they actually project really heavily into the striatum, so the caudate nucleus in particular, and the accumbens, and they also project up to the cerebral cortex in a completely different way to the way that this traditional blue nucleus, this core nuclei, projects. And I think that has really, really important implications for the way that the system is organized. So let's dive into that into a little more detail. So the idea here is that we have these two different populations, one which is really precise and receives precise input from the cortex, and another which is relatively diffuse, is projecting up to a different part of the cortical mantle and is projecting in a relatively diffuse fashion that crosses what we would otherwise call regional boundaries. If people are interested, this is a really beautiful piece of work by Francisco Clasca's group. What this does is exposes the fact that this is not a really true dichotomy. There's a weird and wonderful world of different thalamocortical projections. The ones that I've just described here, the blue one here, the core type, has that projection that comes up and projects into really layer 4, which is the granular layer of the cerebral cortex. The red type, which they've here split into two, is the matrix type, and they come up and they project up and they project broadly to the supragranular regions of the cortex, as well as to the infragranular regions where the cell bodies of layer 5 pyramidal cells sit. And there's also a fourth class, which sometimes blend in with the matrix type, which people call the intralaminar nuclei. and they project really heavily into the striatum, into the amygdala, particularly the central amygdala, and then into some of the inferior layers of the cerebral cortex. And what I want to draw your attention to on the right here is that no nucleus traditionally that we would look at in the thalamus, we would typically sort of partition the thalamus into, say, 30 or so different nuclei. None of them is purely one or the other type. So you can see here that something like the ventrolateral nucleus has a blend of this C type as well as the M type, and then a little bit of the focal M type over here. And so the idea is that the thalamus is not a set of little homogeneous nuclei that are projecting in a very particular way, like cables, you know, that you might wire up in a supercomputer that are all projecting information to one location, but rather is this hub of lots of different types of connectivity that have different implications for how the system can function. So let's just look again at the core thalamus first, because this is an area that has been really, really well studied. Murray Sherman and Ray Gullery did some really, really fundamental work on this circuit. And one of the reasons for that is that it's predominantly the circuit that comprises the parvo and magnocellular pathways of the lateral geniculate nucleus, the main visual pathway in humans and non-human primates and lots of different mammals. And so it was really easy to link its circuit to function. We can go and see whether or not the animal could still see in that particular retinotopic location if we lesioned the nucleus, and we can really pick it apart. And so for that reason, Murray and Sherman came up with sort of two or at least it wasn't exactly them, but their insights really led to two ways of thinking about this system. One of them, it was doing a message passing function, something like a retinal cell got stimulated and then it passed a message to the lateral geniculate nucleus, which then passed it on to the cortex. So something like a relay. If we're being slightly more generous, we might say that they gate. In other words, they have the ability to inhibit a message, or they can pass it on and then quickly quash it. So in other words, they're not just passing a message on, but they can actually change it in fundamental ways. But really, at the end of the day, this circuit is really well set up to precisely contact a target region and to potentially propagate important information through the system. Now, importantly, the matrix thalamus is really not well set up for that at all. The pioneering work here was done by a Kiwi neuroscientist, Ted Jones. And what he showed was that interspersed between all those different core nuclei were these populations of matrix thalamic nuclei that projected up rather diffusely. And so the question is, well, what's their function? It's really hard to kind of follow that message passing concept, that idea, through to fruition when if a signal comes up, it's now immediately distributed out amongst many different regions. And a fact that I didn't mention yet, but the regions in which it contacts the cerebral cortex are actually the places that are chock full of dendrites, which are the parts of the cell that are receiving the inputs, often feedback inputs from higher levels of the cerebral cortex. And so it really raises this interesting question, which is, what is the function of the matrix thalamus? Rather tongue-in-cheek, what I'm going to try to do today is convince you that what it's allowing the system to do is to sort of liberate specialist regions to perform novel coalitions. It's allowing the system to essentially energize, in inverted commas, such that it can function in a particular mode that is beneficial for the effective information processing at the more systems level, rather than the individual level of a particular circuit. So how am I going to try to convince you of that? Well, I'm going to rely on two very brilliant postdocs that are currently working in my lab. Eli Muller on the left has done some great work on what we call neural mass models. So this is taking a real bird's eye view of the system. It's a little bit fuzzy in terms of details, but it gives you a really good conceptual understanding of how the bits and pieces work. And Brandon Munn on the right, who's done some really great work on cellular level models, where they're much more precise, they're much more biophysically realistic, but you lose the ability to really zoom out as much as you could. And in both cases, I think we can really gain some insights into what the benefit of having a system like this matrix thalamus might be. All right. So Eli's project, as I mentioned, was a neural mass model. So what does that mean? What it means is that we make some assumptions about the way that populations of neurons work. Rather than taking individual neurons and trying to track each one of them and trying to say, measure the sodium and potassium within each cell as you have an action potential, or measure the amount of calcium that's liberated when a neuromodulator hits the cell, we instead say, we imagine that we've got a population of these different neurons, let's call them pyramidal neurons, E excitatory neurons, And we assume that the mean of that population, one number, is sufficient to describe their activity. Now, we know that this is not correct. We know that the cerebral cortex has many, many, many degrees of freedom. But it turns out that it's not that bad of an estimate based on the central limit theorem, which says that you've got a big population of unrelated phenomena units. You can basically measure them or estimate them with a Gaussian, and a mean is a pretty good way of tracking the activity of a Gaussian. Neural field models actually look at the variance of that Gaussian, so the width of the Gaussian as well, which can give you a little bit more wiggle room to describe things. So what Eli did was he took a validated neural mass model of the cortico-thalamic system. So you can imagine here on the left, we have a little core loop between a pyramidal neuron with a little interneuron connecting it down to a thalamic core cell, which contacts a little inhibitory reticular nucleus, and then back up to the pyramidal neurons in the cortex. And what he did was he wired them up into what we call a toroidal structure. Looks like a little donut. The reason we do that is that this has no boundaries, right? So that means that there's nowhere where you could fall off the edge of the network and have your activity be meaningless. And what he did was he wired up this network with local connectivity in red, and then we tuned a parameter, the diffuse coupling, which is acting like the matrix system, to ask what does that do to the system's dynamics. And Eli's really brilliant observation and hypothesis was that the matrix thalamic cells are actually doing something a little bit like acting like temperature in a glass of water. So if we took a glass of water sitting on your desk, sitting at room temperature, all of the little water molecules are going to be fizzing around, bumping into each other, but not so slowly that they turn into an ice crystal, and not so fast that they evaporate into gas, but just enough that they're bumping into each other, passing a little bit of energy off here and there. What the idea is that as you heated that water up, one of the things you do is you cause the little water molecules to move faster. And what that actually has the effect of doing is spreading out the distribution of those different water molecules. So as you heat it up, you actually get some parts of the water that are a little bit colder than you'd expect, and some that are actually much hotter. You could think of these as a little bit like delusional water molecules. They think it's hotter than it is, and so they act a little bit more like a gas, and the rest of the system acts a little bit more like a liquid. The idea is that you can imagine automatically then what kind of an effect that would have on the brain. If I start to turn up the temperature of the system, now I'm going to make it such that individual little coalitions of these neurons can jump up into a higher firing rate, can start to have privileged activity and coordination with one another that they wouldn't have had if they were playing around at a lower level of temperature when they didn't have that extra energy. On the right, this is just a way of showing what that looks like in dynamical systems language. The idea would be that at low temperature, the system is subcritical, and as you heat it up and you move towards the right, as you increase diffuse coupling, what you do is you lose that lower line, which we call an attractor, and all of a sudden, the only attractor that's in the system is this line up the top. And so a bunch of regions start to try to drive their activity up to this higher level, but the point of the system being spread out is that the whole system doesn't shift over to that higher attractor. That would be something a little bit more akin to epilepsy. So Eli ran this model. He found the behavior that he was expecting to see, where at low levels of this diffuse coupling, really nothing was happening. And at high levels, the whole system was sort of in lockstep, a little bit like epilepsy. But there was this really fascinating intermediate zone where we saw a blend between these two different states, a state which was quite damped in terms of the reticular nucleus inhibiting the thalamus, and on the other side, a state where the thalamus could now get involved and and activate the excitatory population in the cortex. So in other words, what the matrix thalamus was doing was it's sort of waking up the thalamus and allowing it to actually function and play a role in the system's dynamics. There's really cool functional signatures of this as well. I don't have time to go into them all tonight, but one that I think is quite telling. If you provide a stimulus, so a little pulse response to the network in each of these different zones, and what Eli's done here is he split the network up into the sort of least active third, the middle third, and the most active third in bold. If you stimulate that network, when you do that in the subcritical zone, the activity of that node quickly trickles back down to zero. You can think of it as being really damped. But as we start to move into the quasi-critical zone, the zone which has a blend of these different states, what you actually see is you start to spread out the activity. So now the most active neurons can stick around a little bit longer, and the ones that are a bit quieter, they quickly drop back down to 0. And you can see that if you move even further, you spread that, you really diverge the state out even more. And we think that this is one of the really critical building blocks for really encoding any information in the nervous system. Right? If everything is doing exactly the same thing, you've really got no information in the system. So here on the left, everything trickles down to 0. On the right, everything synchronizes. But in the middle here, what you've got is the ability to distinguish between input A and input B. And we think that's really, really critical for the way that the system can function. I just want to draw your attention to an analogy that I found really helpful for thinking about this stuff. I don't have a background in physics, and I spend a lot of time trying to understand intuitively what really, really brilliant people like Eli and Brennan are talking about. A way to think about this a little bit more conceptually is to think about a ball rolling around a landscape. And if we were over here on the subcritical side of things, this would be a little bit like a ball being stuck in a little valley. No matter how much you kicked it, the ball's just going to keep rolling back down into the hole. But as we turn up that temperature parameter, as we turn up the matrix thalamus, what we're doing is we're flattening out that landscape. In a way, it's sort of like filling the valley with water. Now, all of a sudden, the ball, when it gets kicked, every now and again, can actually get the energy it needs to jump over into another attractor. And now, unfortunately, if you flatten it too much, that actually obliterates the first attractor. Now, this valley over here, which is the synchronous valley, is the only one that lives. So what you want to do is you want to sit between these two zones and sort of shift back and forth as needed, being quiet when you need to, but then moving into a zone where you can start to create novel coalitions when you need to process information. So that's the system's macroscopic level. What's happening at the microscopic level? So what Brandon's really interested in is really detailed neuroanatomy. He comes from a background in visual neuroscience, where they'd go in, look in the thalamus of a marmoset, and they try to understand the spike dynamics of something like the lateral geniculate nucleus in the transition between sleep and wake. And so when he came to me, we were really interested in trying to understand some of the mechanisms of arousal, like the transition from sleep to wake or from awake to anesthesia, but at the really kind of granular level of cells. And we were really inspired by some beautiful work by Matthew Larkham and colleagues in Berlin. And what they've essentially shown is that one of the major cells in the cerebral cortex, this is a layer 5 pyramidal neuron, it's called thick tufted, and it's really, really unique. Now, here's what's unique about it. Its cell body sits down here in layer 5. That's where it gets its name from. And it has a bunch of little connections coming in, which we'll call basal dendrites. And it turns out that these receive the feed-forward connections from lower cortical areas, typically. But then it has this really odd thing. It has this extremely long stalk. It almost looks like a palm tree in a sense, right? It's got this really, really, really long trunk before it then starts to branch out. And then all of these connections up here, the apical dendrites, are where the feedback from a higher area of cortex comes in. And it turns out that there's a little channel sitting on this stalk that it's called a leak channel, HCN leak channel. And so what that means is that if you've got lots of feedback coming in, but you've left the channel open, all of the neurotransmitters that you work so hard to liberate here with your message come down here, and then they leak out this channel, and they have no impact at all on the basal dendrites. However, if you close that channel, now all of a sudden, any feedback that you give it actually causes this really big calcium wave, and it makes this cell here transition from spiking into a burst firing mode. That's like a really, really potent way of communicating. Larkham and colleagues showed quite beautifully that during the awake state, these cells burst like crazy. But in the anesthetized state, you basically are opening up this HCN channel, and you're not allowing any of the communication between these regions. So we were really interested in whether or not modeling up this kind of a phenomena and thinking about the influence the matrix thalamus would have, which is on waking up these apical dendrites, which are up in that supragranular area, what impact that would have on the system dynamics at this more granular cellular level. So what Brandon did was he again put together a really simple model. He's got a bunch of little cell bodies down here in blue. These are the little cell bodies with basal dendrites, and they're all connected to one another. And then he's got this little stalk here, which is like the HCN channel, which is sort of open or closed. And then he's got the apical dendrites, which are receiving the input. And the main thing that he did in the model was change the likelihood that this cell body down here would transition from a regular spiking mode, i.e. with the HCN channel open, to a burst firing mode with the HCN channel closed. We also manipulated another parameter, which is whether or not we had targeted or diffuse input to this apical layer. That's partly due to the fact that some matrix thalamic populations project quite locally. Do you remember those orange ones that I showed you before? Whereas there's other ones that project quite globally. So we wanted to understand a little bit about those differences. And also that the cholinergic and the noradrenergic system in the arousal system both had that similar feature of really local versus diffuse arousal to the rest of the cerebral cortex. So we have these two different parameters now. So in Eli's study, we had one parameter. Now we've got two: the probability of bursting and the spatial. You can think about it as spatial precision of the input from the arousal system or the matrix thalamus. And essentially, what Brandon found, again, I don't have time to really dive too hard into the details here, but it was a non-linear combination of these factors was required to give us a lot of really interesting network level dynamics and network level signatures. So here I'm just showing an example related to some work that I did when I was a postdoc with Russ Poldrak, where we showed that the brain network really integrated and dissolved otherwise kind of community boundaries when you were performing a cognitive task. Well, to relate that to our model, what you would need to do is actually have an intermediate level of bursting. You don't want all the cells to burst, and you need some to burst. You need to be doing something to actually communicate some information. But you need to do it in a way that actually gives you rather diffuse input to the system. If the input is targeted, you really don't get that same network level phenomenon. It actually looks much more segregated, which is a prediction we have for the way that the cholinergic system might affect the brain. So there are lots of other measures that you can look at, some really interesting ones. The Kolmogorov complexity, you could think about this as how many different dictionary terms you need to recreate a time series of spikes. And it turns out that as you increase up here, you need a lot more. So there's a lot more information encoded in the system at the top. And this is a really interesting measure, integrated information, or PHI, which has been popularized by a number of groups around the world, most notably Giulio Tononi. The idea here is that the way that the brain differs from something like a camera, both of them collect information from the world. The camera has little receptors that turn a pixel a particular color based on the amount of energy they have at that part of the where the lens pushes in the information. Whereas in the brain, the idea is that the difference is that we actually do something with the information. We take the information that we receive from the external world and we combine it with internal states and internal information to create new configurations that are informative. And that measure was also really heightened over here on the right-hand side. So we need intermediate levels of bursting and this really diffuse coupling in order to recreate these kinds of features that we think are really characteristic of the awake and cognitively potent brain state. I'm not going to go into the details here, but Brandon extracted some data from an open data set of a monkey. It's intracranial electrocorticography while the monkey's transitioning from sleep to wake, and found that all these three measures all increased parametrically in the shift from sleeping to wake. And then across our brain state, he used a really cool model fitting algorithm, which is it's like a genetic algorithm, to basically work out where in our model space this data could be best fit. And it turned out that the sleep state was really like this low, no bursting mode, but the targeted or diffuse input to the system really didn't matter all that much. But as you started to transition into waking and then into the awake state, what really mattered was that you increase the probability of bursting, not to 1, you don't want everything bursting, you need intermediate amounts, but you have a relatively diffuse coupling. So we think that argues really nicely for things like the matrix thalamus and the adrenergic arousal system, and the role that they might be playing at the cellular level in allowing the system to function the way that it does. All right, so in summary, the thalamus has these so much to tell us. We have been for a long time in neuroscience, have been really thinking about the thalamus as a relay, like the mode on the right hand side, the core nuclei, something like carrying a message. And what I've tried to argue today is that the matrix thalamus is actually really, really important for ensuring that the message gets communicated. Right? You need this ability to combine precision and information processing of precise contents with the ability to actually propagate that information at all. And I think that somewhere in the interaction between these two different modes is a really, really important lesson for neuroscientists about the kind of algorithms we should be looking for when we're trying to explain how the system might function. Now, I'm not going to let you off there because there's still some super cool stuff to tell you. It also turns out that this organizing principle of the core and the matrix thalamus actually extends way beyond the cortex. One part that I want to talk about today, because I think it has really big implications, and I think that as a community, really, we haven't embraced the implications of this vantage point. And the important thing is this: the core and the matrix nuclei of the thalamus almost exclusively receive different input from the basal ganglia and the cerebellum, two huge major subcortical structures in the brain. The core nuclei, particularly in the ventral tier of the thalamus, so the ventral anterior ventrolateral and the medial dorsal nucleus, receive the input from the cerebellum, and the basal ganglia are actually projecting directly to the matrix thalamic nuclei, which is slightly different than the implications of some of the old models that we had, the Alexander DeLong and Strick model of the basal ganglia. So one way that we can kind of, the way we found out about this, I think, is kind of informative, because it gives you an insight into what people have to do to sort of test these hypotheses. It turns out that the core and the matrix nuclei in the thalamus actually express different calcium binding proteins. So they don't actually have any functional role that we know of, but the core nuclei express what we call parvalbumin, and the matrix nuclei express calbindin. There's a third calcium binding protein called calretinin that also expressed mainly on matrix cells, but it's lesser expressed than the calbindin. And some really, really amazing work from a Japanese group led by Kuramoto and colleagues. This was a paper that really, really kind of opened my eyes. What they basically did was they went through in rodents, and they could actually track cells that received inhibitory inputs and cells that received excitatory inputs. So the excitatory inputs are really coming from the cerebellum, but sometimes also places like the superior colliculus and other cortical areas. And the inhibitory zone are really coming mainly from the globus pallidus of the basal ganglia. And it turned out that in the inhibitory zone, these were overwhelmingly matrix thalamic cells. They had calbindin expression, and the excitatory zone were overwhelmingly parvalbumin cells. They had parvalbumin expression. So what that means is that what we can do is we can then go and look at what we know about the functions of the basal ganglia, the circuitry of the basal ganglia, and what we know about the circuitry of the cerebellum. And we can essentially step forward through the network and think, what implication would their activity or their circuitry have on the thalamus, and then again on the cortex with what we know about how they work. So I'll start with the cerebellum. I'm not going to be able to go into sufficient detail, but I thought I'd just try to kind of whet everyone's appetite here. The cerebellum is beautiful and exquisitely organized, much more organized than something like the cerebral cortex. In fact, it looks like a machine in the way that if the cerebral cortex is something like a hedge or a thicket, a bunch of connections every which way, the cerebellum is this beautifully ordered structure. That doesn't make it easy to remember how it's organized, but this is a really beautiful figure from Egidio D'Angelo, that from a really nice Frontiers paper that I think explains really well what's happening. There's input that comes in via these mossy fibers, which come via the cerebral cortex. They come up and hit this granule cells, and the granule cells are absolutely fascinating. They're actually the most numerous cell in the whole brain. They're actually somewhere between 50 to 60% of all the cells in your brain are actually sitting in this really thin layer in the cerebellum, and there's just tons and tons and tons of them. And each of them project up, and they have these really weird looking things running on them, these parallel fibers that look like telephone lines. So those run acrossways. And then there's this really fascinating anatomy where the Purkinje cells, which are the main cell within the cerebellar cortex, they actually sit perpendicular to them in these little slices. They look like squash rackets sitting up, connecting to all of these parallel fibers. And they in turn project down to the major output of the cerebellum, the deep cerebellar nuclei, which then projects right back out to the core thalamus. This fellow over here is the inferior olive down in the medulla. It also projects into the cerebellar cortex and wraps around these Purkinje cells. For those that are interested, there's some really, really cool work thinking about the implication of activating the olive at the same time as the deep cerebellar nuclei, but I'm not going to get into that now. But the point is to say that there's a really, really logical structure in the cerebellum. And one of the really key things it does is it takes inputs and it spreads them out over this gigantic population of granule cells. And you can think about that as doing something a little bit like what the dentate gyrus does in the hippocampus, whereas it creates this like really, really high resolution copy of a low resolution input. Now, algorithmically, people would describe this as sparse distributed representation. The idea is that if you put in a really low resolution copy and then you make a high resolution facsimile of that, let's say, what you've now done is you've sort of really drilled down on the details. So you can now tell it apart from something that was really, really similar to it. You've got this ability, it's almost sort of mapping it in a high dimensional space. And then it has this, this output on the system that comes right back out to the thalamus. And so what I argue in the paper in far more detail is that what the cerebellum is allowing us to do is actually learn really, really skilled behaviors. It's learning particular contexts and then responding to them really precisely in ways that then liberate the cortex to focus on the sort of problem at hand. So this talented child here on the left is balancing, doing sticking and keeping his eyes up, at an imaginary defender. And so he's doing this in a goal-directed fashion, but without all of that feedback from the cerebellum through the thalamus to the cerebral cortex, I think he would really, really struggle to run all of those different parallel processes at once. In contrast, the basal ganglia has a much different architecture. So rather than spreading out the signal the way that the cerebellum does, it actually collapses it down. So there's billions of cells in the cerebral cortex. There's only something like a million or so cells in each of the pathways of the striatum, the indirect and the direct pathway. There's even less cells in the globus pallidus, externus and internus, and then those cells project back to the thalamus, to the matrix thalamus. So in a way, you could think about this as sort of dimensionality reducing rather than dimensionality expanding. And so it's going to have a really different impact. But I think more importantly, it's going to have an impact on the matrix thalamic cells. And I think this is really fascinating. In the past, the way that people would talk about the cortico-basal ganglia system is that the basal ganglia was essentially deciding whether or not a little corticothalamic loop could start to oscillate and then become the functional loop that was, let's say, performing a function, like reaching out to grab a glass of water or imagining what you'll say next in a talk. That would require something a little bit more like the core nuclei, right? Because you need precision and you need to release this to sort of start to function. In contrast, it looks like what the basal ganglia is doing is it's releasing these matrix cells, which means that they're going to come out and they're actually going to spread out the activity. They're actually going to make feedback more relevant and more active. It's going to have the chance of liberating that column, but also columns around it. And I think this has really, really important implications for thinking about decision-making and volitional behavior, and also thinking about what would happen if you started to impair that system in disorders like Parkinson's disease. Now, I realize that I'm well over time, and so I might just very, very quickly jump a couple slides because I think I can just really hammer home this point for you. So the cerebellum, in a nutshell, I think the importance of the basal ganglia and the cerebellum is that they're allowing the cerebral cortex to function in different kinds of modes, right? If the cerebral cortex is running along trying to process information, it's trying to make a prediction and it's trying to propagate a prediction error. Let's say we take that framework and we try to think what would it benefit the cortex if it had a basal ganglia along with it? Well, the idea, if we go back to that idea of the attractor landscape, activating the basal ganglia is going to have the effect of giving you a relatively local flattening of the attractor landscape. That's going to mean that if you squirt in a bit of dopamine and the basal ganglia releases the matrix thalamus, now if you were thinking about maybe having that extra chocolate chip cookie before you go to bed, it's just that much more likely for the ball to roll into that attractor that then gets you over to eat the cookie. In contrast, what the cerebellum is going to do by being able to learn these really, really precise input-output relationships, it's going to kick you along the landscape without you having to do anything. In other words, it's going to create a little rut in the landscape that lets the sort of ball roll around without you having to tell it what to do. So that's the whole reason that the boy can have his balance while moving his stick, is that he's already trained his cerebellum how to respond when he moves in a different direction. In other words, he's coupled to his environment. And I think thinking about things from that systems level perspective is actually a real, really powerful way for us to start to conceptualize the cerebral cortex and the other parts of the nervous system that are sort of imbuing it with some of the many functions that we otherwise ascribe to the cortex just on its own. So I think it's really, really important to think about this system as a whole. I'm not going to dive into those now. I can answer some questions at the end if we want, but I just want to say, this graph is really trying to communicate the fact that we need to think about the brain as a whole system. Just thinking about the cortex is not getting us there. We need to think about the thalamus and the way the thalamus is being recruited by different subcortical systems, and the impact that might have on information processing. And then I once again want to reiterate that Eli and Brennan are just tremendous and did a really, really great job on both those modeling projects. Thank you.
Thank you so much. That was a wonderful talk. I'm going to hand over to Michelle for the Q&A.
Thank you, Max. This is very impressive work. I'm going to check if there is any question. Otherwise, I'll start with my questions because I am a big fan of the thalamus myself.
Oh, great.
So I have like those are mostly questions that are based on curiosity. So often like people are studying the thalamus in terms of its relationship with the cortex or the striatum, and it's made of different nuclei, and there is a sheath of white matter that is going through the thalamus. I've been looking for those papers in the old literature of axonal tracing and everything, and I never found any papers that studied whether or not there is some communication that happen between the nuclei of the thalamus. And if there is some communication between the nuclei of the thalamus, it is definitely way more than a relay station, because it means that you have some local integration. And since you spearheaded like this research on the thalamus and its role in cognition, I was wondering whether you thought about that and whether you were thinking about how to study that.
It's a great question, and I'll just duck out of frame for a second and say that this is the Bible of the thalamus.
I have it. Well, I moved it to my office.
Everything I've learned about the thalamus comes from Murray Sherman and Ted Jones. I had a really interesting discussion with Laszlo Aksady, who I think is one of the world leaders in thalamic anatomy, at a conference in February when we were still able to travel last year. It feels like about 10 years ago, but So it depends on what you mean by communication. It doesn't appear that the thalamus has glutamatergic interneuronal communication. So the same way that the cerebral cortex will send an input in that'll activate a region and activate a region with glutamate and then cause spiking, right? So the nuclei that sit in the thalamus synapse exclusively outside of it. So they'll either synapse in the striatum, in the amygdala, in the cortex, in the colliculus, other areas like that. There is a lot of inhibitory integration. So what do I mean by that? There's a lot of inhibitory cells within the thalamus. There's more in humans than there are, relatively speaking, in primates and so on and so forth. So it's expanded over evolutionary time. And there are many connections wherein a thalamic nuclei will project out of the cortex, but on its way, it'll hit a bunch of inhibitory interneurons, or it'll hit the reticular nucleus. And they do a lot of coordinating. So, and you can have plasticity there. So you don't necessarily have excitatory coordinated activity or excitatory integration, but you do have, I think it's silly to think that they're just off on their own. They're not, it's not like a cell sitting there kind of ignoring everyone around it, right? They're connected to other regions, but they're not doing it in a way that is consistent with what we normally would think about in the cerebral cortex. So this was actually one of the reasons that we argued in another paper that I did with Luca Cocci and colleagues, that the thalamus is actually doing a really interesting thing at the network level, which is actually promoting both integration and segregation. It's promoting segregation by not wanting a excitatory competitor to fire, right? Every time it leaves the thalamus, it sends off a bunch of signals to say, make everyone else be quiet. But when it gets up to the cortex, it projects out and hits many different regions, which will then become more active and which are going to promote more integration. And so it's a really, really interesting role topologically. It's quite distinct from areas, that we typically think about, like the cerebral cortex and things like that, which are probably a little bit more dependent on the kind of information they're receiving rather than their individual circuitry. Hopefully that answered your question.
Yeah. It's well, it's remain an interesting question. Yeah, because I'm always puzzled by the small nuclei that are in the center, the intrathalamic nuclei that are reported as connecting the entire brain, but physically they're surrounded by other nuclei. How can they connect the entire brain and every area in the brain?
Yeah,
such as the centroid one.
Let me go back here to Klasker's work. You would really like if you send me an email afterwards, then I'll send you a book chapter he wrote, because it's so awesome. It's like the implications of the architecture. But so those nuclei you just mentioned, things like the parafascicular nucleus, the centromedian, they're of this green intralaminar type. And while they do project to the cortex, by far they're much more likely to project to the striatum. So their job is really to kind of get the striatum going and to sort of wake it up. We're still really learning a lot about these connections. It turns out that it's actually really, really important for learning. Plasticity of these synapses are extremely important for learning skills. So there's something really fascinating going on with that circuit that's still just starting to get scratched out. The ones that come out to the rest of the brain, or to the cerebral cortex, are really these matrix type and the core type. And the core type are the classic LGN type, and then the matrix type are the ones that come out and hit diffusely. So I think it's a bit of a misnomer to say that the intralaminar nuclei kind of spread out to the whole brain. They kind of do, but not in the way that you might imagine. It's more nuanced.
We have some question in the chat that I'm going to read. There is a question from a physics background from Leah Talosi. She said, never think of the brain as a nuclear power plant described with critical and subcritical states to then burn and create energy and lead to different connectivity states. Have you think in the study about this model similarity?
Yeah, it's a great question. We didn't explicitly think about a nuclear power plant, but we would definitely, in Eli's paper, which was just published just recently, where we found evidence for that, what we call a quasi-critical phenomena. We really were thinking of analogies to energy, in the sort of thermodynamic sense. So at low levels of energy, the system is not doesn't have sufficient energy to overcome its activation energy. It can't move from one attractor to another. But as you heighten the amount of energy, a subset of the nuclei can start to transition. They start to lose that lower attractor. They have a bifurcation, which means that they can jump to a higher level of activity and then have a different influence over the network. Hopefully you don't raise it too much, because that could actually, as you say, cause a nuclear meltdown. So, this to me is an absolutely fascinating area. Before I started, in neuroscience, I'd never heard of the term criticality before. I obviously knew that when you're boiling water, it's a very, very fine grade between when the water is not boiling and when it is boiling, or if you're lowering the temperature when the water goes from liquid to freezing. And that's evidence of something called a critical transition. So a small change in an order parameter, the temperature, has a really, really qualitative change, a really abrupt qualitative change in the state of the system. And it turns out that there's a huge literature in physics thinking about the impact of that kind of architecture to something like an information processing system like the brain. And while there's many, many different ideas about it, one that we are quite excited by is this idea that the brain would sit slightly subcritical. So what does that mean? If there's a little boundary here, where if I turn a parameter, it goes from one state to another, it doesn't actually really help me if I sit right next to the boundary, because then if I accidentally receive a bit of input, I just flip over the other side. But I'd like to be close enough to that boundary so that if I have little changes, I'm still going to get a lot of the benefits of the things that happen near that critical boundary, but I'm not at risk of actually falling over the other side. This is something that's been really popularized by Viola Priestman, but there's lots of different people who've worked on it. And we're really excited by that idea. A lot of the brain imaging patterns that we see fit this signature of quasi-critical or subcritical. And we could see a lot of the benefits of organizing a system like that. And one of the things we're working on right now is that different neuromodulators could maybe tune you to different signatures of that criticality. You can imagine if this is the boundary, if I turned it on its side, you could approach the boundary to the left or to the right. and maybe this is turning up noradrenaline, this is turning up acetylcholine. And so you can get different benefits by moving to different parts of that boundary. So that's a really, really exciting field, right? And it comes back to that first slide where I was trying to sort of say, there's this big, massive, complicated system. And by working with people like physicists, we can start to actually develop a language that can let neuroscientists ask these really exciting questions that, you know, in my training before that, I was just never exposed to these kinds of tools and ideas. And I, you know, wholeheartedly think it's a really exciting area if you're training to kind of get particularly complex systems analysis. Is a really, really great place to kind of really kind of spend some time learning the technology, learning the terms, learning the frame of reference, because I think it's a really, really powerful lens for the nervous system.
Yeah, I should probably update what I'm telling to my students. I used to tell them that the thalamus is a dynamo of the brain. Well, guess what? It's a nuclear power plant now. We have another question from Angelica Zarkali. She's saying, amazing talk, Max. Thank you. Just wondering whether you had planned to look at differential connections, matrix, core, cerebellum to transmodal, unimodal cortical regions. Feel it might provide useful insights.
Awesome. Hi, Angelica. How's it going? Thanks for the great question. So we haven't thought about it structurally. We've done a couple functional studies. We did a resting state study with some 7T data. I think, you know, we were able to replicate the results in 3T, but I really do think higher magnetic strength gives you just so much more confidence in the subcortical structures, signal to noise, when you're looking at it with functional imaging. But yeah, we did a study there where we looked at, we took genetic expression from the Allen Brain Atlas. So it's just six people that that had unfortunately died, that donated their brains to science, and they went through. The group went through and did gene chip analysis at every location in their brain, and then we were able to actually extract out an estimate of how much calbinin expression there was and how much parvalbumin expression, and then we could use those regresses as an index to look at the connectivity in terms of resting state connectivity with the rest of the brain. And we found something like what you asked. The matrix cells in the thalamus were much more strongly connected to the transmodal regions of the cortex. So things like the default mode network, the frontoparietal network, singular percular network. Whereas the parvalbumin enriched regions of the thalamus were more connected into the primary sensory regions, like the visual regions and the somatomotor regions and auditory regions. So we really do think there's something quite important there, you know, lying at the base of it, but we haven't looked at the structural connectivity. Definitely really interesting question. You know, I'm still kind of getting my head around what we can and can't do with diffusion imaging, working with Fernando Calamante here, but not something that, you know, we have in our crosshairs right now.
All right, thank you. I have another question, which is related to evolution. I was wondering whether you were thinking about ascribing your model of the functioning of the thalamus inside an evolutionary perspective. You know, like for some reason you know, the model are saying that we start with these old structures, and then we added layers after layers after layers. So at one point in evolution, the thalamus must have been self-sufficient. But now it becomes a relay station. And I don't know, I just wanted to have your thought about this, if you were thinking to start working on that by looking at comparative anatomy, for example.
Yeah, it's a really great question. It's very close to my heart. My father's an evolutionary biologist, so I feel like in a way it's like the religion of my family. And I always try to sort of scrape things back to an evolutionary perspective. Unfortunately, evolutionary neuroscience is really hard to do. I tip my cap to people that sit in that vocation and do it regularly. And there's some brilliant work that's been done. But comparative anatomy is just a really, really tricky business. For one, you have to remember, I think at the outset, that every species that we have on Earth right now that we can investigate isn't actually like a great example of where the species that we have came from, right? They're the example of what made it and survived in the current climate, but we actually don't really have a great linear trajectory back in time. we do have some ideas about interesting junctions and junction points. And for that, I really lean heavily on Paul Chisek's work at University of Montreal. He's absolutely brilliant. If you don't know his work, everyone should read. It should be like, along with Marcel Messelem, that should be required reading for every young systems neuroscientist. But yeah, in chatting with Paul, we had some thoughts about it, and it turns out that there's this really interesting way to track the evolution of the thalamus. Anne Butler's done some really great work on this, where there's colothalamus and lemnothalamus. There's these different thalamic structures that, oddly enough, through time, it's almost like they sort of fight out this battle. So sometimes in some animals, the lemno will win, and then other times the colo will win. But what it actually looks like in mammals is that the little branch that we came from, the colo, which is actually a little bit matrix-like, had actually kind of diminished down so that it wasn't really working particularly well. And then there was the lemno, it was a really lemno-thalamically, let's say, enriched animal. And then through some, let's say, gene duplication event or something like that, you can speculate all you want. That's one of the benefits of evolution neuroscience. You can speculate and no one can call you on it. The idea is that through a gene duplication event, we may have actually reborn a matrix system, which then came on top of it. And it wasn't constrained by the same rules that may have actually been present for other animals back further in evolutionary time. And so the idea would be that you've had these two systems kind of fighting it out, but the prominence of the matrix thalamic system in our brains and in non-human primates and other mammals might be a really unique feature of the mammalian branch of the tree of life. And it's really, really hard to like go back. Like, we work with some really awesome zebrafish anatomists and a group up in Queensland, Ethan Scott's lab. And we were really excited early on, like, oh man, do they have a matrix thalamus, and does it project to different parts of the tectum, and they've got a tiny little bit of cortex as a project there. And it just turns out in talking to some anatomists that those fish are actually on a completely different branch of the evolutionary tree. And they do something super weird, and it's like nothing like mammals did. And so you could ask the question till you're blue in the face, but the question you're asking is like, the things forked and you're just like looking at two things this way. And it's not clear that you're going to get a really sensible answer about what the function might have been, because the pressures that led to the fish developing that system are just so different than the pressures that led us to develop it. So I hope that wasn't like a really too nerdy of an answer, but I think evolutionary neuroscience is super important, but really, really, really hard.
It's really hard,
to do it really well.
So I just received the message that our time is up. But I really wish we have the opportunity to discuss a lot more. And so, if I'm giving a talk in Australia, that will happen. But eventually, we'll meet again in conference, or we'll invite you to Bordeaux with your family.
That would be amazing.
We'll find a way. We will find a way. So thank you very much for connecting that late to give us this wonderful lecture on the thalamus, and also the cerebellum, the striatum, and the cortex. And the next lecture of the clinical neuroanatomy seminars will be about cognitive neurogenetics on the 25th of March. And every Monday morning, we still have our Neurochino, where we speak about last week best science that got us excited. Well, with this, I thank you all for coming and connecting, and I look forward to see you at Neurochino and at next CNS seminars. Thank you.
Thanks for having me.
Thank you so much. Bye.
Bye.